Each developing lymphocyte generates a unique antigen receptor by rearranging its receptor genes. How are antigen receptors, with an almost infinite range of specificities, encoded by a finite number of genes? This question was answered in 1976 when Susumu Tonegawa discovered that the genes for immunoglobulin variable regions are inherited as sets of gene segments, each encoding a part of the variable region of one of the immunoglobulin polypeptide chains. During B cell development in the bone marrow, these gene segments are irreversibly joined by DNA recombination to form a stretch of DNA encoding a complete variable region. Because there are many different gene segments in each set, and different gene segments are joined together in different cells, each cell generates unique genes for the variable regions of the heavy and light chains of the immunoglobulin molecule. Once these recombination events have succeeded in producing a functional receptor, further rearrangement is prohibited. Thus, each lymphocyte expresses only one receptor specificity. This mechanism has three important consequences. First, it enables a limited number of gene segments to generate a vast number of different proteins. Second, because each cell assembles a different set of gene segments, each cell expresses a unique receptor specificity. Third, because gene rearrangement involves an irreversible change in a cell's DNA, all progeny of that cell will inherit genes encoding the same receptor specificity. This general scheme was later also confirmed for the genes encoding the antigen receptor on T lymphocytes. The main distinctions between B and T lymphocyte receptors are that the immunoglobulin that serves as the B cell antigen receptor has two identical antigen recognition sites and can also be secreted, whereas the T cell antigen receptor has a single antigen recognition site and is always a cell surface molecule. These receptors also recognize antigen in very different ways. The potential diversity of lymphocyte receptors generated in this way is enormous. Just a few hundred different gene segments can combine in different ways to generate thousands of different receptor chains. The diversity of lymphocyte receptors is further amplified by junctional diversity, created by adding or subtracting nucleotides in the process of joining the gene segments, and by the fact that each receptor is made by pairing two different variable chains, each encoded in distinct sets of gene segments. 1,000 different chains of each type could thus generate 10 to the 6 distinct antigen receptors through this combinatorial diversity. Thus, a small amount of genetic material can encode a truly staggering diversity of receptors. Only a subset of these randomly generated receptor specificities survive the selective processes that shape the peripheral lymphocyte repertoire. Nevertheless, there are lymphocytes of at least 10 to the 8th different specificities in an individual at any one time. These provide the raw material on which clonal selection acts. And that's each developing lymphocyte generates a unique antigen receptor by rearranging its receptor genes. Have a great day.